Hi everyone, I hope you are all having safe days now. Today I thought that maybe I should make a video on this topic as there are a lot of misconceptions regarding it. So without further intros, let's get into the topic. The simplest virion consists of two basic components. Nucleic acid, which means single or double-stranded RNA or DNA, and a protein code known as the capsid, which functions as a shell to protect the viral genome from nucleases and which during infection attaches the virion to specific receptors exposed on the prospective host cell. A virion simply means the complete infective form of a virus outside a host cell, with a core of RNA and a capsid. You have to understand the structure of a virus to comprehend how the test works. The COVID-19 home test is a rapid lateral flow antigen test, a type of test that runs a liquid sample along a surface with reactive molecules. The test detects the fragments of proteins of SARS-CoV-2 virus from a nasal swab sample from any individual 2 years of age or older. But on the contrary, this test, like other antigen tests, is less sensitive and less specific than typical molecular tests run in a lab. However, the fact that it can be used completely at home and return results quickly means that it can play an important role in response to the pandemic. Similar to other antigen tests, a small percentage of positive and negative results from this test may be false. Therefore, for patients without symptoms, positive results should be treated presumptively. That is, the person should not be confirmed as a victim of COVID-19 until confirmed by another test as soon as possible. This is especially true if there are fewer infections in a particular community, as false positive results can be more common when antigen tests are used in populations where there is little COVID-19, and the test is authorized for individuals 2 years of age or older, including those not showing symptoms. Let's talk a bit about the PCR test. PCR test, also called a molecular test. This COVID-19 test detects genetic material of the virus using a lab technique called polymerase chain reaction. A fluid sample is collected with a nasal swab or a throat swab, or you may spit into a tube to produce a saliva sample. Results may be available in minutes if analyzed on-site, or a few days or longer in locations with test processing delays. PCR tests are very accurate when properly performed by a healthcare professional. The PCR then uses special chemicals and PCR machine called a thermal cycler, which causes a reaction to occur that makes millions of copies of a small portion of SARS-CoV-2 virus's genetic material. During this process, one of the chemicals produces a fluorescent light if SARS-CoV-2 is present in the sample. This fluorescent light is a signal that is detected by the PCR machine, and special software is used to interpret the signal as a positive test result. This can be helpful to find the disease even in its onset, because this PCR test makes millions of copies of the genetic material, DNA or RNA, of the virus as opposed to the very small numbers of virus in the onset of the disease. The PCR test is regarded as highly accurate. So, that's it for now. I hope you understand the test now. Thanks for watching, and by the way, I would really appreciate if you subscribed, liked and shared. See ya!